Hello everyone, I am Varna here and today I will be taking up on HMP shunt pathway and its significance. So let's see what is HMP shunt, hexose monos phosphate shunt pathway. So today here in this class you will be learning upon the major purposes and significance of this pathway and the reactions coming up and how it is regulated and what are the clinical significance of this pathway. The significance of HMP pathway is a very important one and it is usually asked for the exams. So that is very important. So significance should be studied well. Okay. So let's see the other names of HMP. HMP is also known as hexose monophosphate shunt, then Dickens-Horker pathway, then shunt pathway, then phosphogluconate oxidative pathway and pentose phosphate pathway. These are the other names of this pathway. Usually known names would be like hexose monos shunt pathway, then HMP pathway, then pentose phosphate pathway. And next let's see where it is occurring. It is occurring in the cytoplasm of the cell. And the tissue sites are like liver, RBC, adipose tissue, adrenal cortex, mammary glands, testes and ovary, lens of eyes. So these are the sites in which these HMP shunt pathway is occurring, because these sites are in a great requirement of NADPH. NADPH is required for the proper functioning and maintenance of these tissue cells. NADPH is helping for free radical scavenging. Free radicals are produced during the metabolism. Various metabolism which is occurring in the body is releasing free radicals. These free radicals is danger for the normal functioning of the cells. It would destroy these cells. So it is very important to remove these free radicals. So these free radical scavenging, that is removal of free radicals, is very important, and that is met by various enzymes. These enzymes are dependent upon NADPH. So these NADPH is required in a higher rate in all these tissues for the proper functioning. So that is met by the HMP shunt pathway. So the main objective of the HMP shunt pathway is to yield, it's to give NADPH. Major purpose of the pathway yields NADPH, that is nicotinamide adenine dinucleotide phosphate, to be used in anabolic reactions like fatty acid and steroid synthesis. It is also helping in synthesis of fatty acid and steroids and also for free radical scavenging. Okay. So it's a shunt pathway. Why is it called a shunt pathway? Because it is the substrate is got from various other pathways. So there is a shunting between the pathways. The substrate is coming from some other pathways. So you are familiar with this reaction, that is glucose getting converted into glucose 6-phosphate, that getting converted into fructose 6-phosphate, then fructose 1,6-phosphate. This pathway you have seen. Where have you seen? That is in glycolysis and gluconeogenesis, isn't it? So you have seen this substrates. So this substrate, that is glucose 6-phosphate and fructose 6-phosphate, and this glyceraldehyde 3-phosphate, this was there in the glycolysis pathway. So that is getting shunted into the pentose phosphate pathway. All these substrate, glucose 6 and fructose 6-phosphate and glyceraldehyde 3-phosphate, these are getting shunted onto the alternative pathway, that is our pentose phosphate pathway, HMP pathway. So that is why it is called as a shunt pathway. So let's move on to the reactions, how NADPH is synthesized during the shunt pathway. So let's see the reactions that is occurring in the pentose phosphate pathway. It is divided into two phases. One is the oxidative phase and the other one is the non-oxidative phase. So first we have to see it is divided into two phases, that is the oxidative pathway and non-oxidative pathway. This is the oxidative pathway, reactions occurring in the oxidative pathway and reactions of non-oxidative pathway. And it starts with glucose 6-phosphate substrate starting, the HMP shunt is starting with glucose 6-phosphate in the oxidative pathway. And the rate limiting step is the glucose 6-phosphate dehydrogenase, G6PD, glucose 6-phosphate dehydrogenase, the very first enzyme that is getting involved in the irreversible steps. It is essentially irreversible, and it is the rate limiting step of HMP shunt pathway. So the pathway is divided into two, that is oxidative pathway and non-oxidative pathway. The pathway is starting from glucose 6-phosphate, and the very first enzyme coming is glucose 6-phosphate dehydrogenase, GPD, which is the rate limiting step, and it is essentially irreversible, which comes in the oxidative phase very first. So let's see in detail the oxidative phase, that is glucose 6-phosphate getting converted into 6-phosphogluconolactone with the help of GPD, G6PD, glucose 6-phosphate dehydrogenase, and there is release of NADPH. Wherever dehydrogenase comes, NADPH will be released. So glucose 6-phosphate is getting converted into 6-phosphogluconolactone with the help of glucose 6-phosphate dehydrogenase by the releasing NADPH. Then next, with the help of the enzyme lactonase, 6-phosphogluconolactone is getting converted into 6-phosphogluconate by utilizing a water molecule. And in the third step of the oxidative phase, 6-phosphogluconate is getting converted into ribulose 5-phosphate with the help of enzyme 6-phosphogluconate dehydrogenase, and here also NADPH is released. So in the first and the third step, there is a release of NADPH. First step, glucose 6-phosphate dehydrogenase making the conversion from glucose 6-phosphate to 6-phosphogluconolactone. The next enzyme is lactonase enzyme, by utilizing a water molecule is getting converted to 6-phosphogluconate. This 6-phosphogluconate with the help of dehydrogenase enzyme ribulose 5-phosphate is formed, and here also NADPH is released. So first and the third step of oxidative phase of HMP shunt pathway, there is release of NADPH. Next is the non-oxidative stage of phosphopendose pathway. So in the oxidative state, you have seen that is ribulose 5-phosphate is synthesized, and that is entering into the non-oxidative stage of HMP shunt pathway. This ribulose 5-phosphate with the help of the enzyme epimerase enzyme, ribulose 5-phosphate epimerase enzyme is making a conversion to xylulose 5-phosphate. With the help of epimerase enzyme, it is forming xylulose 5-phosphate, and with the help of isomerase enzyme, it undergoes isomerization. Isomerase enzyme, that is ribulose 5-phosphate isomerase, is acting upon ribulose 5-phosphate, and it is causing isomerization to form ribose 5-phosphate. With epimerization, it is forming xylulose, and with isomerization, it is forming ribose 5-phosphate. And these two with the trans-ketolase enzyme can get converted into sedaheptulose 7-phosphate as well as glyceraldehyde 3-phosphate. And these two with the help of trans-aldolase can get converted into erythrose 4-phosphate and as well as fructose 6-phosphate. And this xylulose 5-phosphate in the initial step of the non-oxidative phase, with the help of this erythrose 4-phosphate with the help of enzyme trans-ketolase can get converted into fructose 6-phosphate as well as glyceraldehyde 3-phosphate. So this is the non-oxidative state of pentose phosphate pathway. Ribulose, it undergoes epimerization as well as isomerization with the help of epimerization as well as isomerization with the help of the enzymes and forms xylulose and ribulose 5-phosphate, and that together with trans-ketolase enzyme can get converted into sedaheptulose 7-phosphate and glyceraldehyde 3-phosphate. That with the help of trans-aldolase enzyme, it is getting converted into erythrose 4-phosphate and fructose 4-phosphate, and the xylulose in the initial substrate is getting converted to glyceraldehyde 3-phosphate and fructose 6-phosphate with the help of trans-ketolase enzyme and erythrose 4 phosphate. So glyceraldehyde 3-phosphate, fructose 6-phosphate and glucose 6-phosphate. All these are the substrates which you see same in that of glycolysis and gluconeogenesis. So that was with the steps of HMP pathway, the steps that are involved in the HMP pathway. So the main objective of this HMP is for the protection of NADPH. Next, let's move on to its significance. How HMP pathway is significant. So the main objective was to yield NADPH. So let's see what is the role of NADPH in our body. So there are various pathways. There are many pathways which is occurring in our body which requires NADPH. It includes fatty acid biosynthesis, cholesterol biosynthesis, neurotransmitter biosynthesis, nucleotide biosynthesis, then also for detoxification, that is reduction of oxidized glutathione and cytochrome P450 monooxygenase enzymes. All these glutathione and also P450 monooxygenase, cytochrome P450 also requires NADPH. So NADPH is required for many pathways for the synthesis, mainly for the synthesis like fatty acid, cholesterol, neurotransmitter and nucleotides. All these biomolecule synthesis require NADPH, and it is also required for detoxification for the removal of free radicals. As I said, free radicals are synthesized in our body during various metabolism, and these free radicals are toxic for the cells. Normal functioning cells will be toxic. So there is a requirement of removal of these free radicals, which is done with the help of cytochrome P450 and glutathione. These enzymes helps in the removal of these free radicals, which is again dependent upon NADPH. So NADPH is required for fatty acid synthesis, fatty acids and various other steroid and neurotransmitter, nucleotide synthesis, and also for free radical removal. So that is the detoxification. It is required for biosynthesis as well as detoxification. Second significant property is free radical scavenging and RBC membrane integrity. Free radicals is formed in cells is destroying the DNA of the protein. So as I said, free radicals is synthesized in our body during various metabolism, and these free radicals is causing damage for the DNA. DNA of the cell is damaged. DNA, proteins, various other protein, membrane. The membrane of the cell is made up of proteins. So these free radicals can cause damage to the DNA or the protein, which is leading to the cell death or cell damage. So that is causing, this free radical is causing cell damage or cell death. So it is important to remove these free radicals. So free radicals is to be removed, and that is done with the help of various NADPH dependent enzymes like cytochrome P450, glutathione. All those are NADPH dependent. So it is important to remove free radicals from the tissues to prevent the normal functioning of the cell. So in case of RBC you all know, red blood cells, it is helping in the transport of oxygen. It is found in our blood. It is helping in the transport of oxygen and carbon dioxide exchange of gases. So that is RBC's function. So RBC cells is getting lysed with the help of with the free radicals. So RBC, when the RBC is lysing, RBC cell is getting decreased. So exchange of gases is not proper. So it is important to maintain the RBC membrane integrity. RBC membrane is made up of protein. So that protein will be affected by the free radicals, which is causing the lysis of RBC. So this NADPH dependent enzyme is helping in the removal of these free radicals, and thus it is helping in maintaining the RBC membrane integrity, which is helping the proper function. It is helping in the proper functioning of RBCs and its normal growth. Example, like glutathione is a tripeptide composed of glutamate, cysteine and glycine amino acids, and reduced glutathione, that is GSH, maintains the normal reduced state of the cell. RBC cells normal reduced state, only in the reduced state it is helping in the normal functioning or the transport of oxygen is done. So reduced glutathione maintains the normal reduced state of the cell, that is helping in the RBC membrane integrity. Next significant is it prevents the formation of methemoglobin. So you all know what is methemoglobin. Methemoglobin is functionless hemoglobin. It cannot carry oxygen. Only if the iron in the RBCs or the hemoglobin, hemoglobin is having an iron, reduced form of iron is present in the RBCs. That is, the hemoglobin contains iron. Only in the reduced state it can carry oxygen. So Fe2 plus is getting converted into Fe3 plus when there is free radicals. So it is getting forming methemoglobin. So if it is not in the reduced state, that is Fe3 plus state, it is in the methemoglobin state. So there is formation of methemoglobin which cannot carry oxygen, and this NADPH is required to keep Fe2 plus form, and thus it is helping in prevention of methemoglobinemia. So NADPH is helping to maintain the reduced form, that is Fe2 plus form of hemoglobin, and thus it is preventing the formation of methemoglobin. In methemoglobin, Fe2 plus, that is the reduced form of iron in the hemoglobin, is getting converted into Fe3 plus, that is methemoglobin formation. This methemoglobin is functionless, it cannot carry oxygen. So if there is more amount of methemoglobin in our blood, means it is called as methemoglobinemia, which reduces the oxygen carrying capacity of the blood. So NADPH is the one which is helping in keeping this Fe2 plus form in the hemoglobin. NADPH is helping in keeping the reduced form of iron, Fe2 plus state. Thus it is preventing methemoglobinemia. Next function is bactericidal activity. Bactericidal activity, that is NADPH is required for the production of superoxide anion radical by macrophages to kill the bacteria. NADPH is required for the production of superoxide anions. Superoxide anions are that which will kill the bacteria. So it is helping in the production of superoxide anion radical, and this anion radical is killing the macrophages to kill the bacteria, helping in killing the bacteria. So it is also involved in bactericidal activity. Next significance is the availability of pentoses, ribose and deoxyribosis, and DNA and or RNA synthesis is done. All these synthesis is done with the help of NADPH. Then next one is the prevention of transparency of the lens of eyes. There is a maximum concentration of NADPH which is seen in the lens of eyes for preserving the transparency of the eyes. So it is there is a maximum concentration of NADPH seen in the lens of eyes, and it says that the transparency of eyes is maintained with the help of this NADPH. Next significance is detoxification of drugs. So various drugs and foreign bodies which is entering in our body is detoxified with the help of cytochrome P450 in the liver. So this cytochrome P450 is again an NADPH dependent enzyme. Cytochrome P450 requires NADPH for its action. So these are the significance, and clinical significance also required. So when you are writing the significance, you should include this clinical significance as well. Clinical significance First and the main one is the GPD deficiency, glucose 6-phosphate deficiency. So this G6PD is a very common one, GPD deficiency. This is mostly seen during clinical practices, and this is an X-linked recessive trait. The defect, this GPD deficiency is transmitted through an X-linked recessive trait, so it is always X-linked recessive. The deficiency will lead to a drug-induced hemolytic anemia or spherocytic hemolytic anemia. So the main symptom is hemolytic anemia, drug-induced hemolytic anemia. So this GPD deficiency is manifested only when they are exposed to these drugs or any toxins. For example, like in anti-malarial drugs like primaquine, and this primaquine can stimulate the peroxide formation inside the RBC. These peroxides can lead to RBC lysis, and those who are GPD deficiency, their level of NADPH will be very low, because NADPH, since this NADPH is very low, the production of NADPH is very low, this NADPH is not available for free radical scavenging or the removal of the peroxysides, which is causing the RBC lysis. So it is leading to RBC lysis. Since GPD deficiency is causing low level of NADPH, this NADPH is not available for removal of peroxysides. Peroxycide, which is formed during the anti-malarial drugs, which is caused by the anti-malarial drug like primaquine. It is stimulating the production of peroxides, and this peroxides is causing the cell lysis, and there is this GPD deficiency. Since there is GPD deficiency, NADPH is also not there to prevent the removal of the peroxysides. So together it is causing cell lysis, and it is causing hemolytic anemia. Anemia means there is lesser amount of RBC. RBC is, number of RBC cells is very low. RBC cell is getting lysed due to the peroxide formation by the anti-malarial drugs. Anti-malarial drugs is leading to the formation of peroxysides, and these peroxysides are causing RBC lysis. So RBC cells is getting lowered. The amount of RBC cell is getting lowered and causing anemia. And also GPD deficiency is causing lower level of NADPH in their body, and then the NADPH is not helping. Since it is not there, it is not helping in the removal of peroxysides, and these peroxysides are actively killing the RBC cells. So it is causing cell lysis. RBC cells are getting lysed here. The ingestion of oxidative agents that generate peroxides or reactive oxygen species. This is like when we are taking these drugs and other toxins, it is causing the release of oxidative agents that generate peroxysides and reactive oxygen species, ROS. All these peroxysides and ROS are causing lysis of the cells. RBC cells are getting lysed because of the production of these peroxysides are causing the drugs like anti-malarial drugs and certain other toxins. They are causing the generation of peroxides and ROS, which is causing the lysis of those RBCs. And also they are having GPD deficiency. So NADPH is not synthesized in our body. NADPH level is very low. So NADPH is also not available for the removal of peroxysides or ROS. So it is not available for detoxification. So the amount is increasing, and these peroxides and ROS are causing lysis of the cell. RBC cells are getting lysed, and it is causing anemia, hemolytic anemia. So that is why it is called as drug-induced hemolytic anemia, since it is caused by the drugs which we are giving. Drugs-induced hemolytic anemia. The drugs are causing synthesis of peroxides and ROS, and this is causing the lysis of the RBCs. And GPD deficiency is causing lower level of NADPH. So NADPH is not available for the removal of peroxides and ROS. So that is not removed, so that is causing the lysis of RBCs. So anti-malarial drugs and purines, glycosides from flava beans, analgetics, then sulfonamide drugs, sulfur drugs are all causing the increased production of peroxides and ROS. So this is the microscopic view of the hemolytic anemia. This is how the RBC cell is getting lysed. This GPD deficiency is also causing flavism. Individuals with G6PD deficiency must not have flava beans. Flava beans should not be eaten by the individuals who are having G6PD deficiency. It can cause acute hemolytic anemia due to the lysis of RBCs, erythrocytes lysis, and it can lead to dark or black urine. Flava beans should not be consumed by individuals having G6PD deficiency. And one of the interesting factor is that the growth of Plasmodium falciparum, that is the malarial parasite. This growth of these parasite fails in G6PD deficient individuals. The geographical distribution of GPD deficiency correlates with the malarial endemicity. The enzyme deficiency offers resistance to Plasmodium infections. The parasite requires reduced glutathione for its survival, which will not be available in adequate amount who is having deficiency of GPD. GPD deficiency is reported from almost all states of India. So those who are having G6PD deficiency, they will not be having malaria. That is one of the interesting point, because the parasite, malarial parasite Plasmodium falciparum, that is in a requirement of reduced glutathione. This reduced glutathione will not be available in patients who are having G6PD deficiency. The parasite requires reduced glutathione for its survival, which is not available in G6PD deficient individuals. So this parasite will not be able to survive in those individuals. This parasite fails in G6PD deficient individuals because it requires reduced glutathione for its survival. Next clinical application is methemoglobinemia. GPD deficient persons shows increased methemoglobin in their circulation. Even though cyanosis is not seen, there will be methemoglobin circulation in a higher amount in their circulation, because G6PD deficiency can lead to increased production of peroxides. Peroxides and ROS is not removed by the NADPH, so it will be in more amount, which can cause to RBC in its reduced state. A reduced state will not be seen. So it is converted into Fe2+. It is not converted to Fe2+, it is in the Fe3+ state, and that is called as the methemoglobin, which is functionless, it cannot carry oxygen. Next one, that is TPP deficiency. It is a trans-ketolase deficiency. It is affecting the trans-ketolase step, which you see in the non-oxidative phase. Trans-ketolase enzyme is deficient. Severe neuropsychiatric symptoms will be seen, lesions and hemorrhages near the third cerebral ventricles will be there. Deranged mental functions and depressions will be seen in such patients. It can occur in Wernicke-Korf's syndrome. The trans-ketolase reaction is measured in RBC as an index of thiamine status in an individual. Thiamine is a vitamin. So the thiamine status will give you the trans-ketolase activity in the individual. The occurrence of the manifestations of Wernicke-Korf's syndrome which is seen in alcoholics and those with thiamine deficiency is due to the genetic defect of the enzyme transketolase. So in alcoholics and also those who are having thiamine deficiency, there will be deficiency of transketolase enzyme. So indirectly the thiamine status of an individual gives the transketolase reactions. The transketolase reaction measurement can be taken with the help of the thiamine status because it is dependent upon thiamine. So those who are alcoholic and those who are having thiamine deficiency will have a defect in transketolase enzyme. Okay, thank you. So this before I close the class, this significance of HMP, including the clinical as well as the NADPH. Functions of NADPH is both important. So when significance is asked, both you have to write, clinical as well as the role of NADPH. Okay, thank you.